Welcome you all. The next point in the unit palynology is the germination and growth of pollen tube. So the germinations of the pollen tube or germination of the pollen that will be start when the pollen tube is formed. So pollen tube that will be formed from the aperture which are present on the outer layer of the pollen tube. From that openings, the tube that will be formed. And this tube is nothing but the pollen tube. So in that pollen tube, all the material which are present in the pollen grain, that is male gametes, then vacuoles, cytoplasm, these all material that will become in the pollen tube. So how exactly this pollen germination will be start? So when the pollen grains that are stick on the stigmatic surfaces. So as you know, at the time of pollination, that means the pollen grains, mature pollen grains, they are dispersed in searching of the stigma. So on the stigma, there is the sticky surfaces which are present in the female gametophyte. So on that stigmatic surface of the carpel is the exactly site of the pollen grain germination. So generally uh, in flower, as you know, at the center there is the stigma, style and ovary, that is female reproductive organ, and in male reproductive organ that is anther and filament. So after the maturations of anther will be breakdown and pollens will be immersed out. So these pollen are, they will be fall on the stigma having the sticky surfaces. That's why they will be attached on that surface of the stigma, and at that time the germination of pollen grain will be start. It is naturally in laboratory, that is in vivo pollen germination. In laboratory, how these pollen germinations will be start? So for study this in vivo pollen germination, the technique that have been used, that technique is known as the hanging drop technique. So this hanging drop technique is used for study the pollen germination. So for this practical, and there is the requirement of the sugar, then calcium, boron are usually required for getting the maximum germination. So sugar solution, calcium, boron, these are the compounds that will be helpful for the germinations of the pollen grains. Then, so what are the exactly functions of that chemical? So first, the sucrose. This sucrose solution that maintain the osmotic potential and they can provide the nutrition to the pollen grain. Then the boron compounds, they will be promoted the absorption and metabolism of sugar synthesis. Also, the pectic material, this pectic material for all elongations and pollen tubes formation that will be helped. So the pollen tube that will be bursting effectively and they can all these controlled by the boron. So here the sucrose that will helpful for providing the nutrition and increasing the osmotic potential. Then boron that will be helpful for the uh, or helpful in the metabolism or sugar synthesis. That's why the pollen tube that will be elongated and this pollen tube will be bursting effectively. Uh, and after bursting, the male gametes that will be enters into the female, or they will be entered to these, or they can enter in the ovary. Then the calcium that will be helping in the pollen growth factors. Calcium that will be binds the carbonyl group in pollen tube wall and giving the rigidity to pectic calcium control the permeability of pollen tube membrane. So this also the calcium that will be helping in the pollen tube membrane, so they can easily give the good rigidity. That's why they can easily enter into the female gametophyte. So these are the three main compounds that will be helpful in the they will be helping for the pollen germination: sugar, boron, and calcium. Then next is time required for germination. So in different plant, the time that will be required for germination, it will be different. So in case of these Lupa cylindrica plant species, in that the time is for pollen tube germination, time required 15 to 30 minutes. In some plant like the lilium, it requires 30 to 60 minutes. So the time required for the pollen germination that will be different in different plant species. According to the pollen nature, the pollen tube germination that will be takes place. Then number of germ tubes. So generally each pollen grain produces either one tube or more than one tube. So of one, only one, if the pollen grain they can produces two or three pollen tubes. In that only one will be effective. Only one do the normal function. Then pollen all proteins. So pollen all, they will be releases certain proteins at the time of germination. So those proteins play a vital role in promoting the germination. These protein outer layer of these pollen grains, they can secreted some these proteins that will be helping in the pollen germination tube. So in pollen germination, uh, naturally the stigmatic surface, it is the site of pollen germination. In that, at the time of pollen germination, the compound like sugar, boron, and calcium, they will be helping in the promoting or absorbing the nutrients, promoting the pollen tube germination. In each plant or the plant that will be required for pollen tube germination, they will be different in different plant species. And in pollen tube, number of uh, at the time of germination, one tube is formed, either two or three pollen tubes are also formed. But in that, only one tube that will be effective, and they do the or they can gather the all material which are present in the pollen grain. Then the proteins that will be secreted from the all of these pollen grains are also important. These proteins that will be helping in promoting the germination. Then next point, it is the pollen tube growth. So pollen tube growth, in that the hydration of pollen grain, first of all the hydration of pollen grains takes place, and in time distinct from regions of the germ pore. As you know, at the outer layer uh, of the pollen grains, they are having some apertures, means some small pores or openings are there. So in that openings, the pollen tube germination that will be formed. So this pollen protoplast, tube nucleus and generative cells enter in the pollen tube. So when after the formation of pollen tube, the material which are present in the pollen grains like protoplast, tube nucleus, generative nucleus or generative cells, they all material they have come enter into the pollen tube. So this protoplasm of the pollen tube produces tube wall material growth of the pollen tube. So this growing zone of the pollen tube is at the tip which is made up by the compartmental cap. This tip zone rich in RNA and some past material and so uh, this tube pollen tube the this growing zone of this pollen tube that will contain some extra material like the RNA, some proteins. So this will be helping to pollen tube to easily fit into the stigma or style ovary. So vesicle formation that will be helps in the tube growth. The vesicles moves along with the pollen tube, pushing protoplasm to the tip. So these vesicles are formed at the tip of this pollen tube, and this will be helpful for the pollen tube for moving along this or pushing the all protoplasmic material which are present in the style. Here, this calcium ion concentration, that is positive ion charge at the tip of tube, creates the growth. So this tube nucleus that will be degenerates, generative cells they will be divided into the two non-motile male gametes. So this formation of the two motile gametes from the generative cells will takes place in the pollen tube. पोलन ट्यूब में हमने देखा जब भी पोलन ट्यूब फॉर्म होती है तब वहां पे ट्यूब न्यूक्लियस और जनरेटिव सेल न्यूक्लियस दोनों भी वहां पे उस ट्यूब में एंटर करते। फिर जब भी ट्यूब में क्या होता है कि ट्यूब न्यूक्लियस है वो डीजनरेट हो जाता है और जो जनरेटिव सेल न्यूक्लियस है उसका डिवाइडेशन होके वहां पे टू मेल गैमेट्स फॉर्म होते हैं। So at that time the pectinase and cellulase enzymes synthesize at the tip of pollen tube and which hydrolyzes the stigma all and nucellus during the fertilization. So at that time when the male gametes are formed, some enzyme that will be also helpful like pectinase and cellulase enzyme, they will be helping for the synthesizing the tips of the pollen tubes. These tubes they will be hydrolyzed, they can absorb the waters from these all of the stigma or all of the style and nucellus, and then the fertilization will be takes place. So at the time of fertilization water will be needed. Fertilization के टाइम पे वाटर की जरूरत रहती है, तब ये जो एंजाइम्स है वो एंजाइम्स जो वहाँ पे सेल्स प्रेजेंट है लाइक स्टिग्मॉल है, स्टाइल है, देन जो उसके साइड में जो न्यूसेलस पार्ट रहता है, they will be absorb the water from that cells and helping this all for the fertilization. Then also there are some factors at that place, they will be affected the pollen tube growth. First is about the stigma surface. So stigma surface, when the dehydrated stigma surface is one of the important factor which affect the pollen growth. The for pollen tube growth, first of all the hydrations will be takes place. So, however, the dehydrated stigma not provides the favorable environment for the pollen grains. जब भी pollen स्टिग्मास्टिक सरफेस के ऊपर गिरता है तब वहाँ पे वाटर जरूरी है, हाइड्रेशन होना जरूरी है, तभी उस उनका पोलन का जो भी है जर्मिनेशन स्टार्ट हो सकता है. यदि जो सरफेस है वो सरफेस यदि ड्राई सरफेस है तो वहाँ पे पोलन का जर्मिनेशन नहीं हो सकता. So the stigmatic surface it is also important regarding the pollen tube growth. Then next is about the sucrose substance. So sucrose substance are required for the osmotic potential as a nutrition during the pollen tube growth. तो जब भी पोलन वहाँ पे पड़ता है तो वहाँ पे सुक्रोज चाहिए, तभी उसका ऑस्मोटिक प्रेशर बढ़ेगा. ऑस्मोटिक प्रेशर बढ़ने के बाद ही वो वहाँ पे जो भी वाटर है वो वाटर एब्सॉर्ब कर पाएगा. तो दिस सुक्रोज substance they are also the important factor in case of the pollen tube growth. So these sucrose substances are present in the pollen grains as well as at the site of pollen germination, that means at the on the stigma. So sometimes these sometimes what happen, the stigma become the non-receptive. Such a non-receptive stigma not provided the sucrose substances for the germination of pollen grain. When the stigmatic surface that will be having the sufficient amount of sucrose, as well as in the pollen there is the amount of sucrose will be there, then and then only the pollen tube growth will be start. If the sucrose concentration is very low, there is no easy, तो ऑस्मोटिक प्रेशर इंक्रीज नहीं होगा. जो भी पोलन है वो पोलन उतनी कैपेसिटी में वहाँ पे वाटर है वो एब्सॉर्ब नहीं कर पाएगा. इसलिए जो सुक्रोज सब्सटेंसेस आर आल्सो इम्पोर्टेंट. Then weather, if the odd weather conditions such as heavy rains, then warm weathers, drought condition, then bright sunlight, high wind velocity, high humidity or low humidity, frost condition are affected the pollen tube growth. And if the heavy rain will be takes place, the pollen cannot easily sticks on the stigma. They can fall down, and pollen growth of the pollen tube it is not possible. If the bright sunlight is there, in bright sunlight these pollen grain are उसका डिहाइड्रेशन हो जाएगा और वहां पे पोलन डेड हो जाएगा, उसमें पोलन ट्यूब फॉर्म नहीं हो पाएगी. सिमिलरली और जो अदर कंडीशन है, ड्रॉट कंडीशन में यदि वार्म टेम्परेचर हाई है तो जो टेम्परेचर की वजह से जो पोलन ग्रेन है, पोलन ग्रेन इजीली ड्राई हो जाएगा. यदि वो ड्राई हुआ तो डेड हो जाएगा, उस वहां पे पोलन ट्यूब फॉर्म नहीं हो सकती. तो दिस वेदर और ऑड वेदर्स आर आल्सो अफेक्टेड ऑन द pollen growth formation. Then pollen sterility. Sometimes the in uneven number of chromosomes they causes the sterility. Such a sterile pollen grains has no capacity to develop the normal length of pollen tube. So if the chromosomal changes or chromosomal number of this chromosome, the pollen will be sterile form. Then this pollen they not generally form the pollen tube, they will be dead. Then pollen incompatibility. Sometimes or in certain times what's happen, the specific fertilization, if the pollen grains of other species falls on the stigma of one species, then pollen incompatibility are occurs. Tubes of specific species does not allow to germinate, and pollen grain will be non-specific species. If the pollen grain of the hibiscus flower, they will be fall down on the rose flower. Then how the pollen tube form or fertilization will be takes place. So each pollen grain having the particular उसी species के ऊपर वो जाना चाहिए, तभी वहाँ पे fertilization होना चाहिए, तभी वहाँ पे fertilization होगा. यदि वो wrong plant species के ऊपर वो pollen गिरेंगे, तब वहाँ पे वो जो है स्टिग्मा है, वो स्टिग्मा पोलन ग्रेन को अटैच नहीं कर के लेगा, वहां पे फर्टिलाइजेशन नहीं होगा. तो इन दिस वे the plant incompatibility will take place. Due to this incompatibility, the pollen tubes are not formed. Then sometimes the style length. If in some plants like the hibiscus, the style length they will be affected the pollen tube growth. Plant style length is more as compared to the pollen tube length. तो जो जब भी पोलन ट्यूब फॉर्म होती है तो पोलन ट्यूब कहाँ तक फॉर्म होनी चाहिए, तो जहां तक ओहरी है ओहरी तक ये पोलन ट्यूब जानी चाहिए. जनरली हिबिस्कस का यदि आप देखोगे तो हिबिस्कस की जो स्टाइल रहती है, स्टाइल बहुत बड़ी रहती है, तो इतने बड़े वहां पे पोलन ट्यूब्स फॉर्म नहीं हो सकते. पोलन ट्यूब फॉर्म न होने की वजह से उस प्लांट में वहां पे फर्टिलाइजेशन नहीं होता है. आप हिबिस्कस के फ्लावर सबने देखे, हिबिस्कस का प्लांट देखा है, तो जो वहां पे स्टाइल है, स्टाइल बहुत लंबी होने के कारण उस प्लांट में फर्टिलाइजेशन नहीं होता है. सो दिस इज ऑल्सो द रीजन or fact that will be affected the pollen tube. Then the boron or calcium concentration. So minerals like the boron or calcium are the important for the physiologically growth of this pollen tube. If they are absent, the not pollen tubes are not formed. If they are having the less concentration, then the pollen tubes are not forming condition. Then trinucleate pollen grains, as you know, in generally in pollen grain there are two nucleus, tube nucleus and generative nucleus. If the trinucleate form, they are goes to the abnormal condition, they cannot be germinate. So trinucleate pollen grains are abnormal pollen grains, and due to this abnormality, pollen tubes are not formed. Then cellulase or pectinase enzyme. If the cellulase and pectinase enzyme they are required for the piercing the stigma all by the pollen tube. जो भी stigma का all है उसे होल वो ब्रेकअप करने के लिए होल वहां से स्टिग्मेटिक सरफेस से अंदर पोलन ट्यूब को अंदर जाने के लिए वहां पे एंजाइमेटिक एक्शन होना जरूरी है. तो यहाँ पे यदि वो एंजाइम दैट इज सेलुलोज एंड पेक्टिनेज एंजाइम वहां पे प्रोड्यूस नहीं होंगे, तो जो पोलन ट्यूब है, पोलन ट्यूब उसके जो स्टिग्मेटिक सरफेस से एंटर नहीं कर सकती. सो दीज आर आल्सो फैक्टर. If the low concentration of cellulose and pectinase are there, then the pollen tube growth will be affected. Then position of the pollen aperture. Generally, in some cases, if the pollen aperture it is above surface, जो पोलन स्टिग्मेटिक सरफेस के ऊपर गिरा है उसका जो होल है वो ऊपर की साइड पे रहेगा, तो जो भी पोलन ट्यूब फॉर्मेशन के लिए टाइम जाएगा वहां पे जो पोलन ट्यूब की ग्रोथ है वो ग्रोथ अफेक्ट हो सकती. यदि वहाँ जो होल है पोलन ग्रेन के ऊपर जो स्मॉल ओपनिंग रहते वो एक से ज्यादा है, तो उसमें पोलन ट्यूब कौन से पोर से ओपन होगी, उसकी जो पोजीशन है पोजीशन भी पोलन ट्यूब ग्रोथ के ऊपर अफेक्ट करेगी. So certain times the tube orientations of the pollen grains on the stigma that will be affected the pollen tube growth. In uniporate pollen grains, if the position of the aperture is opposite to the stigmatic surface, then pollen tube growth not will be takes place. So जो openings है, these opening जो बॉटम साइड के जो सरफेस है स्टिग्मेटिक सरफेस, उसके सरफेस पे ही वो ओपनिंग आना चाहिए, तभी इजीली वहां पे पोलन ट्यूब फॉर्मेशन हो सकता है. यदि वैसे नहीं है पोजीशन, तो दे अल्टीमेटली अफेक्टेड ऑन द पोलन ट्यूब्स growth. So these are some factors that will be affected on the pollen tube formation. So here this point will be over about the pollen tubes growth.